Hello guys. So in this video let's discuss about cell. So class 12 chapter 8 టాపిక్ ఇది సెల్. సో ఈ వీడియో కంటిన్యూస్గా కాకుండా ఐ విల్ మేక్ ఇట్ ఇంటూ పార్ట్స్ లైక్ 4 or 5 పార్ట్స్లో చేద్దాము. సో కమింగ్ టు దిస్ టుడే వీడియో లెట్స్ డిస్కస్ అబౌట్ బేసిక్ ఇంట్రడక్షన్స్ అండ్ బేసిక్ cell discoveries. So coming to the starting introduction, so we divide things into two types, that is living and non-living. సో ఏదైనా ఆర్గానిజం మనకు లివింగ్ అని చెప్పాలి అంటే దట్ ఆర్గానిజం షుడ్ బి మేడ్ బై సెల్స్. సో దట్ ఈస్ ది basic thing which we know. So anything which is made by cell, we call it as living organisms. So hence we call this cell as basic unit of life. Ante to form the life, the basic brick or the basic unit which is needed is nothing but the cell. Now coming to the divisions of organisms based on the number of cells. So based on number of cells, organisms have been divided into two types: unicellular and multicellular. So we already know unicellular organisms are nothing but their body is simple and made by single cell. Multicellular organisms are their body is complex and their body is made by many number of cells. And unicellular organisms lo complete body functions, digestion, excretion, reproduction, everything will be carried forward by a single cell. Multicellular organisms lo each and every function will be carried forward by group of cells. Done. So that is the main difference between unicellular and multicellular. ఇందులో మనకు కావాల్సింది ఏంటి అంటే ఎగ్జాంపుల్స్. సో యూనిసెల్యులర్ ఆర్గానిజమ్స్లో అమీబా, పారమీషియం and bacteria, and yeast and euglena. So these are the main examples of organisms which are unicellular. And coming to the organisms which are multicellular is fungi, plants and animals. So to talk with, so fungi, yeast is a type of fungi, but there is an exception that fungi lo only yeast which is multicellular anamata. Done. So this is the basic kind of organisms divided based on the number of cells. Now let's discuss about the main important point is, so we call this cell as the fundamental structural and functional unit of life. సో దీని మీనింగ్ ఏంటి అంటే, సో ఎనీ ఆర్గానిజం ఈదర్ ఇట్ ఈస్ యూనిసెల్యులర్ ఆర్ ఈదర్ ఇట్ ఈస్ మల్టీసెల్యులర్, సో ఆర్గానిజం యొక్క స్ట్రక్చర్ లైక్ వీ సే లైక్ హ్యూమన్ బీయింగ్స్ చూసుకుందాం. సో వీ హ్యావ్ హర్ హెడ్, నెక్, ట్రంక్ పార్ట్, లెగ్స్ పార్ట్. సో అట్లా డిఫరెంట్ ఆర్గానిజం ఈజ్ హ్యావింగ్ డిఫరెంట్ స్ట్రక్చర్. ప్లాంట్స్ కానీ, యూనిసెల్యులర్ కానీ, మల్టీసెల్యులర్ ఆర్గానిజం. సో ఈ స్ట్రక్చర్ అనేది ఇట్ ఈస్ గివెన్ బై ది సెల్. So hence we call it as fundamental structural unit of life. And we also call it as fundamental functional unit of life. ఎందుకంటే లైఫ్కి కావాల్సిన ఆల్ కైండ్ ఆఫ్ ఫంక్షన్స్ ఫర్ యూ టు లీవ్, రెస్పైర్, వాక్, టాక్, రన్, ఎనీ కైండ్ ఆఫ్ ఫంక్షన్స్కి సో దిస్ సెల్ ఈస్ ది fundamental unit. So that's the reason we call this cell as fundamental structural and functional unit of life. Done. So next we'll discuss about the few important scientists and their discoveries. So first we'll discuss about this. First scientist is Anthony van Leeuwenhoek. So Anthony van Leeuwenhoek is the first scientist who observed the living cells. Done in the year 1660s. So Anthony van Leeuwenhoek living cells ni ela chusadu ante he has collected the rainwater where he could see something moving. Done. So he observed this droplet of water under his own made microscope and he found the cells which are moving. So we call these cells as living cells. Done. So he named, he observed the blood cells and he observed the bacteria. So these are the main observations of Anthony van Leeuwenhoek. And we call this Anthony van Leeuwenhoek as the father of microbiology. Most important point. So this is all about the story of Anthony van Leeuwenhoek who observed the bacteria in the rainwater. So next is the story of Robert Hooke. So Robert Hooke is the scientist who observed the non-living cells. So bark ki unde cork tissue. So he observed the cork cells anamata, so which are non-living generally. So he observed it under his own microscope and he looked, he observed the structure something like this. Hexagonal shaped cells. Then, so he compared this non-living cells, this structure has the honeycomb cells ani he compared it anamata. And he coined or he named the term cell, because cell in Latin we call it as little rooms. Then so if you see the structures it will be looking like little rooms. So hence he named the structures as cell. Cell was coined by Robert Hooke because in Latin we call them as little rooms. Next important term is, important point is, we call Robert Hooke as father of cytology because he was the first person who first identified that these plants are formed of cells. So hence we call him as father of cytology. Father of microbiology is Anthony van Leeuwenhoek. Father of cytology is the Robert Hooke. Done. So now let's discuss about one more scientist. He is Robert Brown. So Robert Brown was the first scientist who observed the nucleus. So he said a circular body which is present exactly at the center of cell, which was named as nucleus. So this was the main important discoveries of important scientists to discuss with in the topic. So first there were simple microscopes. To tell, there were first light microscopes. After that the discovery of electron microscope have been formed. So with the discovery of electron microscope, internal structure of the cells was identified. So presently we see many different kinds of cell organelles, that is due to the discovery of the electron microscope. Done. So this is about the basic introduction of the cell. Yeah. Now let's move towards the most important topic, we say cell theory. So this cell theory includes mainly three scientists which we need to discuss in detail. So let's discuss about the first scientist. He is Matthias Sheldon. So Matthias Sheldon in the year 1838. Done. So in the year 1838, so he was generally an German botanist. So Matthias Sheldon ఏంటి అంటే he observed the different plant cells. So what he said that these cells together will form the tissues. So this was the main observation or main point given by the Matthias Sheldon in the year 1838. Because he was a botanist, he observed the plant cells. Then and he said that cells together will form the tissues. Next comes the second scientist. He is none other than Theodore Schwann. So Sheldon and Schwann అని కూడా అనొచ్చు షార్ట్గా. సో టు ఎక్స్ప్లెయిన్ ఐఎమ్ జస్ట్ ఎక్స్ప్లెయినింగ్ ది ఫుల్ నేమ్స్. సో థియోడర్ షెల్డన్. So in the year 1839. So Sheldon was in the 1838. Schwann was in 1839. So he was a British zoologist. Done. So as he was zoologist, he observed the animal cells. That was his main observations. Now let's see what he observed and what he gave. So he says that every cell has an outer layer which is formed of plasma membrane. Done. This was the first point given by Schwann. Every cell has a thin outer layer which was formed by plasma membrane. And he also observed the plant cells, and he found there is one more unique layer present outside the plasma membrane, and it was nothing but the cell wall. Done. So what he says is animal cells have an outer layer called plasma membrane. Plant cells have an outer layer called cell wall, inside which it will be having the plasma membrane. So animal cells ki they need flexibility. I mean for movement gaani, for growth, to change their shape. So to absorb the nutrients. So these are the things which are needed for animals. So cell wall enti ante it is the rigid wall. So if cell wall is present, animals cannot undergo these all kind of activities. So that's the reason animals don't have the cell wall. Done. So this is two points. Coming to the third point. So he says that bodies of plants and animals are composed of cells and product of cells. So ultimately observation of plant cells, animal cells tarvata he gave that point అనమాట. సో ఇప్పటిదాకా మనం చూసిన షెల్డన్ అండ్ ష్వాన్ థియరీస్లో ఇఫ్ యూ సీ దే గేవ్ దేర్ పాయింట్స్, బట్ దే డెంట్ సెట్ దట్ హౌ ద సెల్స్ ఆర్ ఫార్మ్డ్. అంటే న్యూ సెల్స్ ఎలా ఫామ్ అవుతున్నాయి, బాడీ ఆఫ్ ది ఆర్గానిజం ఎలా గ్రో అవుతుంది అనేది మాత్రం దే హ్యావెంట్ ఎక్స్ప్లెయిన్డ్. సో విచ్ వాజ్ ఎక్స్ప్లెయిన్డ్ బై Rudolf Virchow. So Rudolf Virchow in the year 1855 has said this term called omnis cellula E cellular. So omnicellular E cellular is nothing but cells divide and new cells are formed from pre-existing cells. సో ఇప్పటిదాకా ఏమన్నా వీళ్ళు థియరీస్ ఇచ్చారు కానీ న్యూ సెల్స్ ఎలా ఫామ్ అవుతున్నాయో చెప్పలేదు. బట్ ఇట్ వాజ్ టోల్డ్ బై ది రొడాల్ఫ్ వర్చ్యూ టెలింగ్ దట్ cells divide and new cells formed from the pre-existing theory. So this is about the cell theory. So first is plants and animal cells are made by cells and product of cells. That is your first theory. Second theory is cells divide and new cells formed by the pre-existing cells. So this is the basic introduction of the cells. Now let's discuss about the animal cells and plant cells, prokaryotic, eukaryotic, basic introduction చెప్తాం. So if you see the plant cell where you take the example of onion cell, animal cell where you take the example of cheek cell. So if you see, so onion cell is a plant cell which will be having the cell wall, and cheek cell is an animal cell which won't have the cell wall. Done. So both this plant cells and animal cells will have the nucleus. Done. So it is membrane bound nucleus will be there. Inside the nucleus they will be having the chromosomes, which is nothing but the formed by the DNA and histone proteins. So this is about the plant cells and animal cells. The major difference is cell wall, and plant cells will have the plastids and animal cells won't have the plastids. Animal cells will have the centrosome and centrioles. Plant cell won't have the centrioles and centrosome. So this is the additional point apart from your NCRT అని చెప్పొచ్చు. And next to discuss about the main part of prokaryotic and eukaryotic cells. Let's discuss only about the differences. So మనకు కావాల్సిన ఓన్లీ డిఫరెన్సెసే కాబట్టి, so prokaryotic cells, they won't have nucleus, they will be having the nucleoid, it will be having the nucleus. అంటే ఏంటో చెప్తాం. If this is the cell, this is your genetic material, and if this is the cell, this is your genetic material. So in the eukaryotic cell, genetic material is covered by a layer which we call it as nuclear membrane. If you see in the prokaryotic cell, nuclear membrane won't be there. Genetic material will be freely suspended in the cytoplasm. సో న్యూక్లియర్ మెంబ్రెయిన్ కానీ ప్రొటెక్షన్ కానీ ఏది ఉండదు. జెనెటిక్ మెటీరియల్ నార్మలీ సస్పెండ్ అయి ఉంటుంది అనమాట ప్రొకారియోటిక్ సెల్స్లో. డన్. దట్ ఈస్ ద ఫస్ట్ డిఫరెన్స్. సెకండ్ డిఫరెన్స్ ఏంటి? ఐల్ కంటిన్యూ ద లైన్ లైక్ దిస్. సెకండ్ డిఫరెన్స్ ఇస్ prokaryotic cell won't have membrane bound cell organelles. Cell organelles. So eukaryotic cells will have the membrane bound cell organelles అంటే mitochondria, Golgi complex, endoplasmic reticulum, ఇవన్నీ కూడా will be there in the eukaryotic cell. Next, both the prokaryotic and eukaryotic cells will have a fluid like substance or jelly like substance which we call it as cytoplasm. So in the cytoplasm all the cellular activities will be occurred. Done. This is the one more common point anamata. Next we'll discuss about few cell organelles. So first is the ribosomes. So ribosomes is non-membrane bound. So non-membrane bound organelle కాబట్టి it is present in both prokaryotes and eukaryotes. So location ఏంటి అంటే it will be present in the cytoplasm. In the plant cells it will be present in chloroplast, and It will be also present in the mitochondria. It will also be present in the rough endoplasmic reticulum. So this is the location of the ribosomes. Next is you have centrosome. ఇప్పటిదాకా చెప్పింది. So it will be present only in the animal cells, and it is also non-membrane bound, which help in cell division అన్నమాట. సో సెల్ డివిజన్ టైంలో మైటోటిక్ స్పిండిల్ ఫైబర్స్ రిలీజ్ చేయడానికి ఇట్ ఈస్ రెస్పాన్సిబుల్ ఫర్ మైటోటిక్ స్పిండిల్ formation. So this is about the basic, and in later videos prokaryotic cells, cell organelles gurinchi will be discussing in detail. This is the basic introduction anamata ante. This is an introduction class. Now let's discuss about few cells, their shapes and their sizes. Very important straight questions adigi chances untayi. So first is your mycoplasma. So mycoplasma is the smallest known cell, which varies to 0.3 micrometer in length. So that is about your mycoplasma. Next comes your bacteria, 3 to 5 micrometer. Next is your egg of ostrich. So ostrich unfertilized egg is the largest one anamata. So egg yolk cheppali ante it will be having the surrounding white part giving the nourishment, shell untundi giving the protection. So yolk part will be the largest anamata. So that is your largest, and smallest is mycoplasma. So if you see the RBCs, it will be round, biconcave. Then the size will be 7 micrometer in diameter. And let's see the nerve cell. So ostrich cell largest one, and don't confuse between this, that's the reason I'm underlining. Nerve cell is the longest one in length anamata. So it is the longest cell, which is also branched anamata. Then that is about the nerve cells. And if you see WBC cells, it is amoeboid in shape. And if you see columnar epithelium, so this columnar epithelium will be long and narrow. It will be something like this, itla long ga narrow ga untayi anamata, and it will be having the hair like structures. So that is the structure of columnar epithelium. Next is you have tracheid cells in plants. They are elongated as usually. And next you will be having the mesophyll cells. So mesophyll cells are round and oval. So round and oval cells. So this mesophyll cells lo they will be having the chloroplast anamata. So plant cells lo untayi mesophyll cells. Generally upper and lower epidermis ki between lo you can see the mesophyll cells which is having the chloroplast. So it will be round and oval shape. So this is all about your this video introduction about these cells, where I have covered most of the topics. And so what I will tell is, in the upcoming videos I'll discuss about prokaryotic cells, eukaryotic cells, and if possible, we'll also discuss about the PYQs అనమాట. సో ఇన్ ద అప్కమింగ్ వీడియో ఆన్ దిస్ స్పెసిఫిక్ కంటెంట్, ఇవాళ ఈ వీడియోలో డీల్ చేసిన స్పెసిఫిక్ కంటెంట్ మీద విల్ గో త్రూ MCQs అండ్ అసెషన్ రీజన్ కైండ్ ఆఫ్ క్వశ్చన్స్ని కూడా విల్ గో త్రూ దట్. సో హోప్ యూ లైక్ ద వీడియో.